So good evening. I'm Marcia Moore, and on behalf of the Gateway Science Museum, I am so pleased to welcome you to this, the final talk in our 7-part Fall 2020 Museum Without Walls lecture series. Before proceeding to tonight's presentation, I do want to thank our MWOW committee, our Executive Director, Adrienne McGraw, Exhibits Curator, Stephanie Parker, and Community Board Member, Dr. Rachel Teasdale. We also are extremely appreciative of all our speakers who have donated their time, talents, and knowledge to the furtherance of teaching and learning about science in our region and beyond. We also thank our series sponsors, North State Public Radio Station, Renee and John McCamus, and all of you for coming and spreading the word about Gateway and these talks. And we do acknowledge and are mindful that CSU Chico stands on lands that were originally occupied by the first people of this area, the MWOW, and we recognize their distinctive spiritual relationship with this land and the waters that run through campus. We are humbled that our campus resides upon sacred lands that once sustained the MWOW people for centuries. Tonight, we are delighted to welcome Dr. Troy Cline, Associate Professor in the Department of Biological Sciences at CSU Chico, whose topic, Slamming the Door on a Pandemic, COVID-19, and the Science Behind Vaccine Development, is of current and vital interest. Dr. Cline received his PhD from Ohio State University in 2009, where he worked on understanding the relative importance of a newly discovered immune factor on protecting against respiratory virus infection. From 2010 until 2013, Troy joined Dr. Stacy Schultz-Cherry's lab at St. Jude Children's Research Hospital in Memphis, Tennessee, where he published papers on the 2009 pandemic H1N1 influenza virus and on why some strains of bird flu cause more severe disease than others. Finally, in 2013, Dr. Cline joined the Department of Biological Sciences at Chico State, where his research focus has shifted towards understanding the risk that regionally circulated avian influenza viruses pose to humans and commercial birds. Students in Dr. Cline's lab are currently researching the features that determine waterfowl susceptibility to influenza virus infection. And briefly, before Dr. Cline begins, I will remind you that there will be a Q&A session at the end, and we'll ask you to use the chat format for your questions. And now, Troy.
Thank you, Marcia. And thank you, everybody, for showing up, especially the many students that I see in the participants list. I'm flattered and kind of overwhelmed by the participation here. There are so many of you, and I hope that I'm up to the task. So I'm also very appreciative of the forum that the Gateway Science Museum provides local experts for engaging the community on these topics that are of great importance for our region, but also the entire country. So I do want to add in a little conflict of interest statement here. I'm not important enough to have a financial or professional conflict of interest in any of the specific vaccines or vaccine strategies that I might mention tonight. So I just wanted to throw that out there. I will show some data related to one of the vaccines, but it's not because I have any particular interest in this company or this specific vaccine. So what are my goals for this evening? First, I hope that everybody here learns something new. At least 2 or 3 things in a talk about a rather complex issue that might seem a little bit dense at times. I hope you can pull 2 or 3 things away that you can make part of your knowledge, and that you can use that new knowledge in making informed decisions for yourself and for those in your sphere of influence. And then I also want to increase your confidence in the scientific process. And hopefully what I say will set your mind at ease that, at least in terms of the strategies that are being employed to develop a vaccine for COVID-19, you can be assured that the best science available to us is being used to develop this vaccine. And then lastly, because this pandemic is an ongoing situation and new information will continue to come out after I speak tonight, I'm going to end by providing some resources that you can continue to learn more about this topic and related topics so you can stay up to date on how the situation is changing. And I also hope that you'll take the opportunity to share those resources with others so that the information here can go beyond the relatively small number of participants that we have on this in this talk. So a brief overview of what I'm going to cover. First, going to introduce a couple of actors in the play that we're talking about. The first being the virus that we're trying to protect against, that's SARS-CoV-2. Then I'm going to introduce our immune system briefly, how the immune system works, sort of a big picture bird's eye view as it pertains to vaccine immunology. I'm going to talk about several different types of vaccines that have been used in the past, pros and cons of each, and some examples as well. And then we'll finally arrive at our destination and we'll talk about some development, some strategies specifically that are being used to develop a vaccine against SARS-CoV-2, how are vaccines tested, and where do some of our current candidates stand. All right, so I want to start by throwing out a couple of scenarios. And as I throw these out, you can see them on the screen already, I want you to think, would you prepare for these situations? And how would you prepare for these situations? So the first one is a big exam. Right, this is real life for some of you students. It's happening right now. How do you prepare for a big exam? What about a job interview? How would you prepare for a job interview? And those are exciting enough. But the next one we're going to talk a little bit more about because it's more exciting. How would you prepare for the this will never happen to any of us here, but how would you prepare to face Clayton Kershaw in an important at-bat in a high-stakes game? And I apologize for any Giants fans who are stinging a little bit today as they watched their mortal rival win a World Series last night. I'm from the East Coast, so I'm neutral to this rivalry. But how would you prepare for that? How do people prepare? We would probably all prepare rather poorly, but people who can play the game of baseball, how do they prepare to face Clayton Kershaw in an important situation? What they do, maybe you'll know this, is they'll sit down in the team's facility and they'll watch hours and hours of film on Clayton Kershaw. They'll watch everything about how he throws the ball, his release point, the mechanics of his windup. They'll study all of his tendencies. What does he throw in the first pitch of an at-bat? What does he throw if there's nobody on base? What if there's someone on third base? What if nobody's out? What if two people are out? What if he's seeing you for the first time in a game or the third time in a game? They'll study all of his tendencies so that when they come to the plate in that high stakes situation, they'll have an idea of what to expect. So they'll be prepared and they'll increase their likelihood that that confrontation will end successfully for the batter. All right, we're going to maybe come back to this scenario in just a bit, but let's move it into biology. How would you prepare for the risk of exposure to a pathogen? And I want to make sure that we're all on the same page about what I mean by pathogen. I'm talking about a microorganism that can cause disease. Could be a bacterium, could be a virus, could be a fungus. How would you prepare for the risk of exposure to a pathogen? What is the infectious disease equivalent of sitting down and watching hours of film on Clayton Kershaw? Right. Maybe you know where I'm going with this. We'll come back to that. But let's talk just for a second about the pathogen that we're here to hear about. It's a virus, SARS coronavirus 2, SARS-CoV-2. SARS stands for severe acute respiratory syndrome. SARS-CoV-2 is the virus that causes the disease COVID-19. These are very specific terms. They're not to be used, well, they often are used interchangeably. And my kids know that I huff and puff when I hear that. But they're not interchangeable terms. One's a virus, one's a disease. As all viruses, SARS-CoV-2 needs to get into a host cell in order to replicate and make more copies of itself. That's the purpose of all viruses, if they have a purpose. All viruses really are genes covered up with protein and some fatty molecule called a lipid. Sometimes they have lipids, sometimes they don't, but it's this protein and lipid shell around a genome. And for SARS-CoV-2, that genome is made out of RNA, which is related to DNA, which is the genetic material for all living things on Earth. So it's that genome. It contains all of the instructions for making all of the proteins that the virus needs to make, as well as for making new copies of the genome. I want to point specifically to one of the proteins that's important for vaccine development, and that's the blue protein that's projecting away from the virus particle in this cartoon diagram. It's the spike protein, or the S protein. For SARS-CoV-2, this is the protein that's important for getting the virus into that host cell so it can replicate. So going back to our analogy, what is the infectious disease equivalent of sitting down and watching hours of film on Clayton Kershaw? It's vaccines. In this situation, it's our immune system that has to be trained on how to deal with a potential pathogenic threat. So if we present a dead or weakened version of a virus to our immune system, cells of our immune system will recognize that dead or weakened version of the pathogen. Alarm signals will get sent, and those cells will respond. And of particular importance for an effective vaccine, we want our B cells to produce these pathogen-fighting proteins called antibodies. And we want our B cells to be able to remember what that pathogen was, so that if it's ever exposed to the real deal virus in real life, it has a memory of it and it can recall that response and protect you before infection becomes established, before you become sick. So I want to make sure that we all remember what antibodies are. And another word for the thing that antibodies bind to is antigen. That's a more specific, more precise term. Antigens are what antibodies are binding to. These are foreign substances that trigger an immune response. And for SARS-CoV-2, the antigen that we care about is the spike protein. Right? There's solid evidence that the spike protein is what our B cells respond to predominantly when we're infected naturally. And in order to be protected by a vaccine, we have to train our B cells up in how to recognize that spike protein. So if it sees it in real life in the context of a virus infection, we're ready to respond. Okay, we know vaccines work. There's all kinds of evidence that vaccines work. But I think very simply, we can just look at diseases that used to exist at much higher rates in our country prior to vaccines being licensed for these infectious diseases, and where we stand now with the incidence of disease. So vaccines were named by the CDC as one of the 10 leading public health advances of the 20th century, right up there with decreasing tobacco use and infant and laboring mother mortality rates. So you can see so many of these diseases, right? If you are of a certain age, maybe you had some of these diseases when you were a child, but your kids probably didn't, your grandkids wouldn't have. It's because we have vaccines for these, and you can see the incidence of disease has increased 95 to 100%. These are diseases that we just don't see anymore in the United States and in much of the world, and that's due in large part to immunization. So the ability of a vaccine to protect an individual upon subsequent exposure to that pathogen depends on how well it does these three things. Our immune system has to detect the antigen. That detection stage is mediated by cells, types of white blood cells that are called dendritic cells or macrophages. They'll take up that foreign antigen, and then those cells will move into lymph nodes. B cells reside in the lymph nodes. They'll see that antigen and they'll get activated. Dendritic cells will show that antigen to T cells. Right? T cells are really picky. They need to be shown that antigen, and that will happen. And then the T cells become activated. The B and T cells then leave the lymph node and they learn how to respond. They learn what is an effective response against that foreign antigen. And importantly for vaccines, we need those B and T cells to retain a memory of that antigen. So if they see it again in the context of an actual infection, they know how to respond, and they'll recall that response and protect us from disease. So this is really what we're trying to do. We're giving our immune system a practice fight with like a crash test dummy version of a virus, so that when they see the real virus, it knows how to respond to protect us. Okay, so we've talked about SARS-CoV-2, we've talked a little bit about our immune system. Let's start talking now about some of the ways vaccines can be developed. I'll talk about, what do we have here? 1, 2, 3, 4, 5. We've got 5 different strategies here, and I'll talk in a little bit of detail about each of them, and not in the order that they're presented here. But first we have DNA and RNA vaccines that code for the antigen of interest. We can have vectored vaccines. We can have whole virus vaccines, either of the killed or inactivated variety, or the live attenuated variety. Attenuated means weakened or wimpy. And we can have the protein or subunit based vaccines. Regardless of the strategy, there are advantages and disadvantages to each. But regardless of the strategy, the goal is the same: to train our immune system on what that antigen looks like, so that it has the ability to recall an effective protective response when we are infected, if we are infected. So let's start in the middle of that chart on the previous slide and talk about the whole virus vaccines, and we'll start with the live attenuated vaccines. So what the vaccine is you're presenting the antigen of interest in the context of the whole virus, but it's a weakened version of the pathogen that won't cause disease in nearly every case. One of the advantages to these live attenuated vaccines is they induce a more robust immune response than the alternative inactivated vaccines. That's because you're delivering the virus. It's going to go through 1 or 2 rounds of replication, and so your immune system sees it in a more natural state, and you get a more robust immune response. Now, one of the disadvantages, however, is that if you are in a category of people who would be considered immunocompromised, you are at slightly elevated risk of contracting disease, even from this weakened version of the pathogen. So what are some of those conditions, those states that would be called immunocompromised? If you're too young, or if you're on the other end of the age spectrum. Pregnancy puts a woman in a state of being slightly immunocompromised. So because of the slightly elevated risk, if you're in one of these states, you would avoid being given one of these kinds of vaccines. Some examples are the oral polio vaccine. We don't use that in the United States, but it is a live attenuated polio virus. The influenza virus nasal spray. So if you get the flu mist, my kids got the flu mist a couple of weeks ago, whereas I got jabbed in the arm, but my kids got the flu mist. That's a live attenuated virus. The inactivated vaccines are the whole virus, but they've been inactivated in some way, treated with a chemical solvent, heated to inactivate the virus. They're more stable, so that's an advantage, especially in parts of the world where maybe the cold chain is difficult to maintain. So they're more stable, and there's no risk if you're immunocompromised. Okay, so if you've got a 10-month-old, the 10-month-old can get the inactivated shot. If you're 65 years old, you can and should get your flu shot. You usually need multiple doses because these vaccines aren't as good at triggering an immune response. So that can be a disadvantage depending on how easily it is to develop or to to deliver multiple doses of a vaccine. Now, for the flu shot, it's just one dose, but for some of the inactivated vaccines, it's more than one dose. So the injected polio vaccine, that's what we use in the United States, and the flu shot, if you roll up your sleeve and get jabbed in the arm, that's the inactivated influenza vaccine. The vectored vaccines are next. These are, it's a whole virus vaccine, I suppose, but in this case, you take a completely unrelated virus to the virus that you're trying to protect against. You strip away most of its genetic material and you replace it with a gene that codes for the antigen that you're interested in. So for example, if we wanted to make a vectored vaccine that would protect against SARS-CoV-2 infection, we take some unrelated virus, strip away its genes, put in the gene for the spike protein, and then you inject that. The advantages here are that viruses are already nature's perfect little gene delivery system. And so by using a whole virus, you are delivering a gene efficiently, and you can produce a lot of the antigen of interest, so you trigger a strong immune response. One of the potential disadvantages is if the virus that you select as your vector, as the backbone for this vaccine, if that virus is one that you've been exposed to in the past, you'll already have antibodies against it, and those antibodies might attack and remove that vector vaccine before it has a chance to deliver its payload. So you've got to be careful about which virus you choose as the vector. Some examples here are vaccines that probably nobody here has received. The Ebola virus vaccine, which was just licensed by the FDA and approved by the World Health Organization last year, is a vectored vaccine, as well as a dengue virus vaccine. The trade name is Dengvaxia, and that's a vectored vaccine as well. And then we've got the protein-based or subunit vaccines. This is, you're stripping away everything except for the antigen of interest. You inject only that part of the virus that is going to induce a protective immune response. So this would be like purifying the spike protein of SARS-CoV-2 and injecting that, removing it from the context of the rest of the virus. Advantage, safety. Disadvantage, that antigen that you're injecting might not mimic what would normally naturally be expressed during a natural infection. And so the immune response that you raise against it may not actually protect against the real life antigen when you see it naturally. But we do have vaccines that are these protein-based or subunit vaccines, and they're actually really effective, and they are among our safest vaccines, the hepatitis B vaccine and the human papillomavirus vaccine that your preteens get. And then lastly, we've got those DNA and RNA vaccines. And I want to back up here just a bit and introduce you to a little bit of molecular biology 101. You know what DNA is. You know the DNA is the instructions for making proteins, right? And the proteins determine all of our inheritable traits. Metabolic traits, our appearance, all of these things are determined by the proteins. Well, DNA doesn't just go right to being protein. There's another molecule in the middle called RNA, messenger RNA. So DNA gets converted into messenger RNA, and then the messenger RNA is used to make the protein. So that's what DNA and RNA is. So you could develop a vaccine where you're injecting either the DNA that codes for the antigen of interest or the RNA, right? So skip the DNA to RNA step, just put the RNA in, and the protein will get made, and your immune system will respond to that antigen. Advantages, there seems to be something about, these are like the IKEA vaccines. I've heard them referred to as the IKEA vaccines because you're just giving the instructions and your cell and the parts, and your cells put everything together. But there's something about that process that actually engages both the B cell and the T cell arm of your immune system. And when B cells and T cells work together, the overall immune response is much better. In fact, when the T cells help out a little bit, the B cells end up making better antibodies than they would otherwise. Just in terms of manufacturing the vaccine, there are some big advantages to these, because once the technology and the platform is developed, it's easily scalable and it's easily transferable to another pathogen. Once you've got the genetic information from some new pathogen, you can quickly and easily develop a new DNA or RNA vaccine. All right, disadvantages. This is kind of new technology. There aren't any DNA or RNA vaccines licensed currently for use in humans. So maybe we don't know what some of the disadvantages are yet. There is a DNA vaccine that's used in horses, and it's been used for like 15 years, but it protects against West Nile virus. Okay, so those are the types of vaccines, and all of those strategies are being employed to try to develop a SARS-CoV-2 vaccine. So let's talk about how vaccines are tested. You should know, and we're going to focus just on the human trials, clinical trials, but you should know that vaccines don't move to the human clinical trial phase until studies have been done in cell culture, right? So just cells in a petri dish, and if possible, in animal models of disease, right? So there's some preclinical data that's collected on safety and if there are any signs that the vaccine would even be effective before a company or an institution can apply for a clinical trial. The clinical trials occur usually in 3 phases, and the goal overall is to establish safety and efficacy. So in phase 1, usually fewer than 100 people are recruited to participate in a phase 1 clinical trial. And really, you're just testing out your vaccine at multiple doses. And you don't even care at this, you care, but you're not looking at this point to see if it shows any signs of efficacy. You just want to know if it's going to hurt people or make them sick or cause any severe side effects. So phase 1 trials are usually small, and they're usually in healthy individuals. So you're not going into the elderly population or the pediatric population for a phase 1 trial. In a phase 2 clinical trial, slightly larger pool, you're still looking for safety, of course, and you're beginning to look for early signs of effectiveness here. You'll draw blood from your patients and see if they're making antibodies against that antigen. Is there evidence of an immune response? And then if everything's looking good, you can go into a phase 3 clinical trial, and this is large scale. Just the recruitment phase of these trials usually takes a long time. We're into the tens of thousands of patients, 30, 60, 70,000 individuals recruited for these trials, and you're testing safety again, because sometimes the adverse events associated with a vaccine are very, very rare. And so you're not going to see those adverse events unless you have a large test group. So this is where you can start to see some of those less common adverse events if they're going to occur. And then you're also testing efficacy. Now, after a vaccine is licensed and used, safety is continuously monitored. There are a couple of federal systems that collect and store data on adverse events associated with vaccines. So let's take a little bit closer look at how phase 3 clinical trials play out. You've got your 30 or 40 or 50,000 patients recruited to the trial. You're going to give half of the group the vaccine. You're going to give the other half of the group a placebo control, right? And hopefully, in order for this to work and be useful, both groups have to be similar, right, in terms of sex, ethnicity, age. So you need similar groups. Usually, you track these 2 groups for about a year, and you're just looking for who gets infected. Typically, companies doing vaccine trials wait until they have about 150 to 200 events. All right, they wait until 150 to 200 people in their trial get infected before they start analyzing the data. Sometimes they do some interim data analysis with a smaller number of events, and that's happening for some of the COVID-19 candidates. right? But if you've got 200 events in our little imaginary trial here, and 50 of them are in the vaccine group and 150 of them are in the placebo group, well, that's evidence that the vaccine's protecting people. Right? So if you get about a 50%, you want it to be as high as possible. But a vaccine that has about a 50%, a protection rate of about 50% in the vaccine group, you can start to work with that. So where are we with some of the COVID-19 vaccines that are being developed? You can see all of the strategies that I already talked about are being used. There are about 180 vaccine candidates in some stage of development, and about 40 of them have moved into some level of human testing. This graphic down here is a bit outdated. It's from late May. But you can see even as of late May, some of these early vaccine candidates had moved into those phase 1 and phase 2 clinical trials. And we know now that there are several of them that are now into the phase 3 clinical trials. And people are anxiously awaiting the release of some of that data in the next month or two as these companies begin to do interim analyses on their phase 3 clinical trials. So what kind of data are we looking at? Like in a phase 2 trial, say, or you're drawing blood from those patients and you're asking, are there signs of an immune response in that blood? Well, you're looking for antibodies, particularly what's traditionally been thought of as an effective antibody response is a neutralizing response. That's where the antibody is going to bind to the virus and prevent that virus from getting into a target cell. So you can get that data from a phase 2 study. And so what I want to do is show some of the phase 2 data that was published in the New England Journal of Medicine. This is, so you may have heard of the Moderna vaccine. That's kind of why I selected this one to talk about, because it's in the news a lot. The Moderna vaccine is an mRNA vaccine coding the spike protein. And so here's their study design. They have 45 individuals. They broke those 45 people up into 3 different groups of 15 people each. And each group of 15 got a different dose of their vaccine, 25, 100, or 250 micrograms. They actually got that dose twice, 28 days apart, right? So 25 micrograms in the arm, 28 days later, another 25 micrograms, right? They're going to be measuring safety, reactogenicity, and immunogenicity. So are we triggering an immune response? So they're going to draw blood from the patients and they're going to look to see if there are antibodies and if those antibodies are functional. Both of those things are important. So, okay, scary data, right? Lots of bars and numbers. But let's just take it a little bit at a time and break this down. So the 2 panels of this data up top, what the authors of this paper are looking at is, are there immunized patients? Are there antibodies in the blood? In C and D, they're asking, are those antibodies functional? Right. So maybe that seems a little counterintuitive. If there are antibodies, of course they're protective, but no, not necessarily. You can have antibodies that don't really protect all that well. And so you got to demonstrate protective effect as well. Right. So it's like my kids go to the refrigerator and say, there's no food in here. And I say, sure, there's plenty of food. Well, that's not what I want, right? There's food, but it doesn't work for them. Okay, but what you can see simply here, just by the fact that the bars climb up the y-axis in all 4 of these panels, and without regard to what dose of vaccine was given, this research team is detecting antibodies that bind to the spike protein. And they're showing that those antibodies, if you mix those antibodies with virus, and then throw that virus onto cells and ask the virus to replicate in these cells, the virus won't. The virus gets neutralized by the antibodies. Okay? We're doing this outside of an actual human body, right? So that may not translate into whether the antibodies would protect from natural infection. That's why you immunize a bunch of people then set them out to live their lives in a phase 3 trial and see if they get infected or not. But this is good data. This is promising data. When Moderna released this, published it, and other companies with other vaccine candidates have released similarly promising data. This was all very good news. So we're hopeful. But there are a lot of unanswered questions, such as, will the vaccine offer long-term protection? We know from related coronaviruses that cause common colds that the protection that you get, well, we don't have vaccines for the common cold coronaviruses, but the protection that you get from natural infection isn't long lasting. And so there's kind of this excitement about the data that's been published, but this hesitance to get too excited because we don't know yet if the vaccine will protect in the long term, or will you only be protected for 6 months? Will the vaccine be effective in those belonging to high-risk groups? What if you're 70 years old and the vaccine doesn't work in you? Well, that's the problem, because those are the people who are at highest risk. Right? Diabetes, high blood pressure, underlying lung complications, right? So if those people don't respond as a demographic to the vaccine, then there's a challenge to slowing down this pandemic. Are there ethnicity-related differences in efficacy? There's been some concern expressed by Dr. Fauci that some of the clinical trials for some of these vaccine candidates haven't included a broad swath of our population in terms of ethnicity. And without those data, can we be sure that we're going to be protecting everybody? Will a single dose be sufficient? Maybe of these 3 things, this is the least important for us, but it's what we need to know. Are you going to need just 1 shot, or do you need to get back for a booster? And then maybe the most important question of all, will people have confidence in it? Will people take the vaccine? California has joined with Colorado, Nevada, Oregon, and Washington in forming a Western Pact, committing to an independent review of an FDA-approved vaccine, right? With the potential that we get a vaccine, the Western states are going to have an independent review by doctors and scientists, that's good, but it's also a bit unfortunate that the whole thing's become a bit politicized, and we're taking these other steps as a precaution. All right, so my last thing I want to do is provide these resources. And Marcia, I don't know if you share, I know the recording will go on the website, so you can, I don't know, maybe this is where, if you're at home, maybe you want to take a screenshot here. But these are some helpful resources. The National Museum of Natural History has a Science of Vaccines lecture series. These talks are from, you know, the real experts, the men and women who are right there doing vaccine science. They are a little bit more nitty-gritty, down into the weeds and the details. So maybe this talk will offer an introduction to those more specific talks. The Johns Hopkins University Coronavirus Resource Center tracks cases nationally and globally, mortality rates, testing rates, anything you want to know. You could lose yourself for hours in that website. If you want to follow the pandemic locally, Butte County, I saw Linda's on the call here. You guys are doing a great job at Butte County of providing a website that is intuitive and accessible. I follow it religiously every day. I'm checking the cases in Butte County, mostly because I want my kids to stay in school. The state of California, COVID-19.ca.gov, also has a good website. You can keep track of this tiered, colored tier system that we have for reopening our economy safely. And then I can't say enough about TWIV, This Week in Virology. This is a general virology podcast by some guys at Columbia University and University of Michigan. And there are weekly episodes, and it's on all things virology since February, nearly every episode. And it's been more than weekly, so it's well over 100 episodes now, all about SARS-CoV-2, any aspect of SARS-CoV-2 that you're interested in. I walk to work frequently and listen to this as I walk. It's fantastic. Clinicaltrials.gov. If you are interested in seeing if there's a clinical trial near you that is recruiting patients, right, either for vaccines or for therapeutics, go to clinicaltrials.gov. type in your information and see if there's a vaccine trial near us that you might volunteer yourself for, okay? But whatever we do, follow the science, right? Scientific problems have scientific solutions. So we need to keep it with the science and go get your flu shot. Won't protect you from SARS-CoV-2, but it will keep those hospital beds open for the patients who need them the most. Thank you, and I'd love to take your questions.
So great.
That's great.
And I
Thank
think
you. Go ahead, Marcia.
as people are coming up with their questions, I just want to thank you very, very much for such a clear presentation. And I thought you were going to, with the baseball player, say we should be spending our time, instead of arguing with each other about masks and so on, informing ourselves in this way. So thank you for helping to inform us. Rachel, you may proceed.
Thanks. Thanks, Troy, very much. That was really interesting, and I really appreciate you taking the time to share this with us. It was very informative. I want to remind everybody that if you have questions, please enter those in the chat, and I would be happy to share those with Dr. Cline so we can hear his responses. So there is already one question in the chat, and that is, could you please give us some examples of some types of safe viruses that are used to carry the vector of SARS-CoV-2?
Yeah, so there's some peer-reviewed published information on, at least in animal models, with an adenovirus. It looks like adenovirus, but it's adenovirus. Adenoviruses, they can cause all sorts of diseases. Many of them are respiratory viruses that cause common colds. And there are a lot of different types of adenoviruses, right? So there is always the risk that, somebody who gets the vaccine will have already been exposed to the specific adenovirus that you're using as the vector. And then maybe they wouldn't take well to the vaccine. Not that it wouldn't be safe, but they wouldn't be protected maybe. But we can do sero studies. You can look through the literature and see, if you want to choose this particular adenovirus, what do we know about the prevalence of antibodies against that adenovirus in the human population in a given geographic location, to get an idea of whether it would make sense to use that vector or some other vector. The cool thing about the adenoviruses is that they naturally infect respiratory epithelial cells anyway. So it's there at the site of your lungs that you're getting this immune response induced, which is helpful.
Okay, thanks. So another question is, if people who are infected with, or not infected, but who are exposed to, or their immune system is exposed to these vaccines, if that sort of trains their immune system to respond, then why are people, maybe it's just anecdotal, but why are any people getting the COVID-19 more than once? Wouldn't their immune systems already be trained now to fight it?
Yeah, you'd hope so. So what we know right now is anecdotally, right? There have been one or two or a handful of cases where people are confident enough that what they're seeing is a true reinfection. Earlier in the summer or in the spring, there was some talk of potential for reinfection, but at that point, people didn't know if it truly was a reinfection. It turned out it was their negative test that they had received was a false negative. But there is some evidence now that some people truly seem to be getting reinfected. So, and that raises some questions for whether the vaccine will be effective. But with just a handful of cases, it's really hard to say, right, at a societal or national level, whether the reinfection rate is going to be significant and whether it will significantly impact our efforts to have a vaccination program. It's also not clear in those people who seem to have been reinfected whether they really, maybe they've got some underlying compromised state that's undiagnosed or undetected. And in those individuals, they didn't respond to the natural infection very well in a protective way. So I think right now it's just too early to tell if the reinfection rate is critical.
Okay. And then maybe related to that is, based on what we know now, do you think that once the vaccine is available, is it like a lifetime vaccine, or is it something where you think there may need to be periodic revaccination? Possibly
there would need to be periodic revaccination, but not for the same reason as we need annual influenza vaccine. For the influenza virus, you know, it can mutate from year to year, and the strains that circulate evolve away from the strains that are in the vaccine and the protection that we've been given through prior vaccination. Coronaviruses don't seem to do that. We know a lot about coronaviruses from studying common cold viruses, and those coronaviruses don't seem to mutate with any significant rate. And so if revaccination is required against SARS-CoV-2, it's likely to be because the protection, the antibodies that are induced in response to the vaccine are not long lasting. Right, so yeah, it's possible that you'll need periodic revaccination, but for a different reason than for the flu shot.
Right, okay. Another question that came up is, are the trials at any stage testing people who are immunocompromised? Or are we only testing, like you said, that there was some concern about the demographics of the population who are participating in the trials, but it seems like some of those demographics of concern would also be how a vaccine might help or potentially harm somebody who's immunocompromised.
Yeah, boy, I'm not certain. I know for sure in the phase 1 trials, you're really steering away from anybody that you think is in a group that may respond adversely to the vaccine. I don't know. I don't know if they steer away from immunocompromised individuals. I mean, some people, you may be immunocompromised and not know it.
Yeah.
So for Moderna's vaccine, for an RNA vaccine, I can't see why that would be a problem, right? Getting the RNA vaccine isn't going to cause safety issues, but if you're immunocompromised, you may not respond to the vaccine, or so you might not develop antibodies and show signs of having been protected.
Okay.
Vaccine, but it's only for potentially those whole virus live attenuated vaccines that it would be an issue.
Right.
I would imagine if that's the vaccine you're developing, then you're going to stay away from putting that into immunocompromised people.
Mm-hmm. Okay. So if the virus were to mutate, would it change the spike protein that might make some of the subunit vaccines less effective than the RNA-based vaccines?
Possibly. So again, we can learn some things that we know from the other coronaviruses we can apply to this question. The spike protein, because it sticks out there away from the virus particle, I mean, that's a juicy meal for our antibodies, and the virus knows it. And so evolution this happens for flu, evolution drives the flu has a different, it's not called the spike protein for flu, but it plays a similar function. The analogous protein for influenza viruses changes in response to our antibodies. The common cold coronaviruses, they really don't seem to do that. There's really no evolutionary selective pressure, it seems, driving significant mutations in those spike proteins. There is one little mutation that's starting to show up in SARS-CoV-2, and it's now the dominant strain that's circulating. But that mutation does not show any significant or convincing signs of meaning anything in terms of whether it escapes from our antibodies, whether it is causing more severe disease, or whether it's transmitting more quickly. So it's interesting, it's something to follow, but the mutations that have been seen in SARS-CoV-2 as it circulates they seem to be neither here nor there. Viruses mutate, that's what they do, it's expected. And this virus isn't mutating any faster than one would expect. And it does not appear that the mutations are impacting disease severity or transmission.
Okay. So in a sort of practical sense, if younger people are less likely to have significant or severe illness, will it be important for them to receive the vaccine once it's available?
Yeah, because the evidence all points to, you're right, young people don't get as ill, it seems, fortunately, but especially the preteens and teens, they can get infected just as easily as an older person, and they can transmit the virus with equal efficiency. So even though they're not getting sick, in order to protect the community at large, it's very important that they're immunized as well. Same thing for influenza virus, right? If you're 12 years old and healthy, the chances of you getting sick, really sick from influenza virus is really low. But that 12-year-old can transmit it to grandma or grandpa, where more severe illness can occur.
Okay.
So everybody needs to be immunized, or as many people as possible need to be immunized in order to protect the whole community.
I see. And in terms of sort of demographics of the way the virus affects people, is there a simple reason why some demographics are more affected than others by this virus? We know there's associations with low income, socioeconomic factors and access to healthcare, but are there other factors?
Sure, yeah. So I mean, biologically, physiologically, people who are older tend to have already kind of suboptimal or lessening respiratory function. So in those patients, if you have severe inflammation occurring in the lungs, the airways are going to be more frail and fill up with debris and fluid, and you'll get the associated pneumonias, which pose a risk to life. But heart conditions as well. I'm not quite as sure on heart conditions. Diabetes is a bit of an immunocompromised state, but the heart conditions as well lead to more severe disease. And I'm not sure about the mechanism of that.
Okay. And along those lines, will there be a need for a different vaccine for people who are immunocompromised or the elderly?
Possibly. So there's some precedent for that. There are a couple of vaccines that are licensed for Streptococcus pneumoniae, your pneumococcal vaccine. And based on your age, whether you fall in these different risk categories, it's recommended that you receive one vaccine or the other. And so if there are, those vaccines don't fall under the category of any of the vaccines that I talked about earlier, but it certainly is something that occurs, that being assigned to a risk group or being of a certain age may preclude you from getting a certain type of vaccine.
Okay. And so having multiple organizations looking for vaccines might help facilitate finding the right one for.
Oh, sure. Yeah. It's actually good that we've got so many different strategies being employed. So, I mean, if all we ended up with was a live attenuated vaccine, maybe your most at-risk group, your older people, wouldn't even be able to get that vaccine, right? Because the live weakened vaccines can cause disease even in older people. So, yeah, we'd be in kind of a bad situation if all we ended up with was the live attenuated vaccine. So it's a good thing that all of these different strategies are being used, and hopefully more than one of them comes through for us.
Right. And is there any concern that the Moderna vaccine, being a nucleic acid vaccine, is there any concern that would be the first one used in humans?
I don't think so. Yeah, I mean, I guess there are some real advantages to having an RNA vaccine. But like I said, some of the disadvantages may be the unknowns, right?
Mm-hmm.
Dick Cheney's unknown unknowns. It's a real thing. I mean, but personally, I don't have any more concerns about the RNA vaccine than I do about the other strategies. It's unprecedented to have an RNA vaccine, and this one, they're trying to make it at like record pace, right? To get this vaccine developed and out really quickly. And so maybe, you know, not having the typical amount of time that would be required to go through these clinical trials. Perhaps the vaccine is released, and then over a year or two, as it is administered, maybe we learn that it isn't as effective as we had hoped for, or maybe we would begin to see some safety issues with it. Hopefully not, but there are a lot of unknowns with that vaccine.
Right. Okay.
The science behind it is solid, and the technology behind it is solid, but it's difficult to know yet what we don't know about that vaccine.
Right. Okay, and maybe this is a good wrap-up question. Can you tell us what you know or what you think about ways the vaccine might be distributed once it is available? How will they decide how it's distributed, who it's distributed to? I don't know if you have insights about the cost, or who would pay and who wouldn't have to pay, or those kinds of things.
I don't have any special insights into that. How the vaccine is distributed is less a question of science and more a question of logistics and transportation, shipment. I mean, what we've heard from the president and leaders in the government is that the goal is to have this, at least I think this is what we're hearing, the goal is to have a vaccine when it's available, available to eventually to whoever would need it without cost to them. And I think that's the way to go.
That would certainly get more people vaccinated than otherwise, I suppose.
Absolutely, absolutely will. Thank you, Dr. Abruzzo, for correcting my my memory of the unknown unknowns. It was Donald Rumsfeld, not my
All right. Okay. Well, Dr. Cline, thank you so much for this really interesting, important, and very useful information. We really appreciate your time putting it all together and sharing it with us. And we just can't thank you enough. We really appreciate it. So, Marsha, Adrian, I think you might have a final thought.
Definitely add my thanks, and I really, this has been inspiring. It's been great to see so many questions, and you have left us all with a framework for evaluating what we are going to be reading over the next year about the vaccines as they come out. And that is our job, is helping prepare citizen scientists. Even if it's a field that is not well known to us, we can all learn, and you are a great teacher. So we really thank you very much. And in response to your question, I think this can be on our website in just hopefully the next few days. Adrian will facilitate that, but it doesn't need any editing. And so if people didn't get the pictures, or if they want to go back and review some of the things that you said, it was so well done. I would recommend that to everybody. And I will then say once again, thanks to everybody for coming. We are definitely a museum that is in operation, maybe not full operation. We are going to be having talks like this throughout the year. Remember that these are usually in October and April, and we're already planning for our April series, but we may have some pop-up Museum Without Walls lectures. So keep us in mind, go to the website, and make sure you're on our email list and share information with your friends and family. Adrian?
And I would just add, like us on Facebook and follow us on Instagram, and that's where we post a lot of things as well. And just wanted to thank everyone for being here, and thanks to Marcia and Rachel for all the hard work that you did to put our series together this time around, and I look forward to the next one. Thank you so much, Dr. Cline. We really appreciate your time.
Thank you. Thank you very much.
Oh, and vote.
Yes. Vote and get your flu shot.
Oh, yeah, definitely.
Both.
Got my flu shot over a month ago. Bye, everyone.